Chromosome analysis or karyotyping. This test is performed to help diagnose genetic diseases, some birth defects, and certain disorders of the blood and lymphatic system. This test elevates the number and structure of a person's chromosomes in order to detect abnormalities. Chromosomes are thread-like structures contains the body's genetic blueprint. Each chromosome contains thousands of genes in specific locations, which are responsible for a person's inherited physical characteristics, and they have a profound impact on growth, development, and function. The sample required for this test includes a blood sample drawn from a vein in your arm, or a sample of amniotic fluid or chorionic villus from a pregnant woman, or a bone marrow or tissue sample. Humans have 46 chromosomes, present as 23 pairs. 22 pairs are found in both sexes, autosomes, and one pair, sex chromosomes, is present as either XY in males or XX in females. Normally, all cells in the body that have a nucleus will contain a complete set of the same 46 chromosomes, except for the reproductive cells, eggs and sperm, which contain a half set of 23. The type and degree of the problem may vary from person to person, even when the same chromosome abnormality is present. Some chromosomal disorders that may be detected include Down syndrome, Edwards syndrome, Patau syndrome, Klinefelter syndrome, Turner syndrome. Interpretation of test results must be done by a person with specialized training in cytogenetics. Some findings are relatively straightforward, but others may be very complex, so further testing techniques may be performed to further investigate chromosomal abnormalities.
Well, all chromosomes are not exactly like one another. They come in types depending upon their structure or shapes and functions. The first basis for classification is shapes. We know this is the structure of a typical chromosome. And is this a single chromosome? That's right. The difference is that this chromosome is not duplicated, while this one has a duplicated copy attached to itself. So these are the duplicated sister chromatids which are attached to the same chromosome. Now, to understand the types, let's first understand the typical structure. Here, these two regions are called the arms of the chromosome. This arm is named as the P arm and this is known as the Q arm. So how do we recognize which is the P and which is the Q arm? Is it based on their positions? Nope, it depends on the size. The smaller arm is the P arm while the larger one is the Q arm. So this chromosome has one P and one Q arm, while this one has two P and two Q arms. Now let's focus on this chromosome structure. Can you guess what this structure is called to which the arms are attached? Well, this knot-like structure is called centromere. It's the portion of DNA that acts as a link between the two sister chromatids. But that's not its sole purpose. Its major function is the attachment of spindle fibers during the cell division process. And let me tell you one more interesting use of the centromere. It's the position of the centromere that helps us categorize the chromosomes into 4 types. The 4 types include metacentric, submetacentric, acrocentric, and telocentric chromosomes. Let's have a look at the structures of each type. Aren't these simple to understand? This one is called metacentric type. As we can see, the centromere is placed almost exactly at the center. The two arms seem almost of the same length. We can say that the centromere is placed somewhat in the middle of the complete structure. Now the next type is the submetacentric. Here, the centromere is placed slightly away from the center. As a result, the two arms of the chromosome appear unequal. The next type is the acrocentric. In this, the centromere is seen almost towards the end region. The term acro in Greek relates to peak. Thus, the name acrocentric refers to the type where the centromere is near the terminal region. Lastly, what we see is that the centromere is present at the extreme end. That is, the centromere is placed at the telomeric region. Hence the name telocentric. However, these chromosomes are not found in humans. The human karyotype lacks telocentric chromosomes. But what is karyotype, you may ask? Well, it's one way of representing all the chromosomes found in a cell. This is how chromosomes are classified on the basis of their structures or shapes, to be precise. Now let's have a look at the other type of classification. This is a much simpler type of classification. Here, the chromosomes are classified whether they are autosomes or sex chromosomes. Now what could these be? This is a representative image of a typical human karyotype. The chromosomes that pair from 1 to 22nd are called autosomes. That is, they are not the sex chromosomes. To be precise, they have got no function to deal with the sex of the individual. The last pair, however, determines the sex of the person. So if the two are identical, that is, both are X and X, then the individual is a female. And what if both are different? In such a case, when one chromosome is X and the other is Y, the individual is a male. This pair is the sex chromosomes. So the chromosomes which help in determining the sex of an individual are sex chromosomes, or even called allosomes. The others, which are from 1st to the 22nd pair, are not the sex chromosomes. This makes them the autosomes. Both contain genes that code for proteins which make up our body. Now tell me, what will happen if there are any alterations in its structure? And what if the total number of chromosomes get altered? Let's find the answer in the next part. This is a representative karyotype of a human. By now we know what a karyotype is. Can you try to give me some conclusions of it? You may say that it looks perfectly normal. But let me spot a flaw for you. Do you notice this set? This is the 21st chromosome set. Instead of having a normal pair of chromosomes, here we find a triplet. Now you may say that it may be good for the individual as it has an extra chromosome. But that's not the case. Any change in the number of chromosomes creates a lot of defects in the normal functioning of the body. On this note, let's understand chromosomal mutations. As the name says, chromosomal mutation means the mutation of chromosomes. And how exactly does it get mutated? It can happen in two ways. One way is where the structure of the chromosomes gets disrupted. The other is where the number of chromosomes are different than the usual in the organism. Let's have a look at each type. This is the structure of a typical chromosome. Now imagine this small part gets physically removed. This would result the chromosome in getting smaller as its segment is lost. This also means that if the segment had some important genes, then they will also get lost, which can result in severe abnormalities. Such type of a mutation is called deletion. This is where the part of the chromosome gets deleted. Now imagine this part of the chromosome gets duplicated. As a result, the chromosome's length increases. Needless to say, this will also result in some or the other abnormality in the individual. This type of mutation is called duplication. Now comes the next type. These are two different gene segments. Imagine they get flipped. Thus, the chromosome will appear like this. This is called inversion. Do you think this type of mutation is harmful? It may or may not be harmful depending on which genes get flipped. Now let's take one more chromosome. Imagine a part of this chromosome gets removed and gets inserted in this chromosome. What will be the result? That's correct. Both the chromosomes will change drastically. This type of mutation is called as insertion. And now comes the last type. Instead of a segment getting inserted to another chromosome, let's imagine a situation where a segment from each chromosome gets interexchanged. So this segment will get into this chromosome and vice versa. This kind of mutation also results in abnormalities as both the chromosomes get altered completely. Any idea what this type will be named as? Well, this type of mutation is called translocation. All these mutations resulting in the disruption of normal chromosomal structures are broadly capped under a single heading. They are categorized as chromosomal aberrations. Now, this was about the mutations that involve changes in the structure of the chromosomes. What about the mutations that involve a change in the number of chromosomes? Let's understand that in the next part. This is a normal chromosome, while this is a disrupted one. When the structure of a chromosome gets altered, it's called chromosomal aberration. We have seen this. Now let's move on to the second type. It's the mutation that involves a change in the number of chromosomes. It's called as aneuploidy. In literal terms, aneuploidy means the presence of an abnormal number of chromosomes in a cell. So the karyotype of a person with trisomy 21 is an example of aneuploidy. The presence of one additional chromosome in the 21st set gives rise to a total of 47 chromosomes in a person. So instead of a normal 46 chromosomes, this person has an additional chromosome. To be precise, an additional autosome. This case of aneuploidy is called trisomy 21. This is because there are 3 chromosomes instead of 2 in the 21st set. The problem of trisomy 21 leads to a condition called Down syndrome. We will get into the details of these disorders in our higher grades. But for now, let's focus on understanding the chromosomal mutations. This aneuploidy involved an autosome. Now tell me, what if the number of sex chromosome alters? So instead of XY, what if an individual has XXY? Is that even possible in the first place? Well, yes. There can be individuals with an additional X chromosome. This condition is called Klinefelter syndrome. Such individuals exhibit both masculine as well as feminine characteristics. Now, in the case of females, imagine that instead of addition, there is a deletion of an X chromosome. This would mean XO. So this individual will have only one X chromosome. This condition is called Turner syndrome. There are a few developmental abnormalities in such scenarios. These individuals have a total of 45 chromosomes only. These were a few examples of aneuploidy that involve autosomes and sex chromosomes.